immature dendritic cells persist in the peripheral tissues for variable lengths of time. When an infection occurs, they are stimulated to migrate via the lymphatics to the local lymphoid tissues, where they have a completely different phenotype. The dendritic cells in lymphoid tissue are no longer able to engulf antigens by phagocytosis or by macropinocytosis. However, they now express very high levels of long-lived MHC class I and MHC class II molecules. This enables them to stably present peptides from proteins acquired from the infecting pathogens. They also express very high levels of adhesion molecules, including DCSIGN, as well as high levels of B7 molecules. They also secrete a chemokine that specifically attracts naive T cells. This chemokine, called DCCK, is expressed only in dendritic cells in lymphoid tissues. These properties help to explain dendritic cells' ability to stimulate strong naive T cell responses. Although activated mature dendritic cells will also present some self-peptides, the T cell receptor repertoire has been purged in the thymus of receptors that recognize self-peptides presented by dendritic cells, and thus T cell responses against ubiquitous self-antigens are avoided. In addition, tissue dendritic cells reaching the end of their lifespan without having been activated by infection also travel via the lymphatics to local lymphoid tissue. Because they do not express the appropriate co-stimulatory molecules, these cells induce tolerance to any self-antigens derived from peripheral tissues that they display. The signals that activate tissue dendritic cells to migrate and mature after taking up antigen are clearly of key importance in determining whether an adaptive immune response will be initiated. These signals can be generated through direct interactions with pathogens or by cytokine stimulation, but in both cases they are thought to be a consequence of the recognition of invading pathogens by non-clonotypic receptors of the innate immune system. The best understood example is the response to gram-negative bacteria, whose cell walls contain lipopolysaccharide LPS. Receptors that recognize LPS are found on dendritic cells and macrophages, and these associate with the toll-like signaling receptor TLR4, which then activates the transcription factor NF-kappa B. Signaling through the pathway induces the expression of B7 molecules and the cytokines such as TNF-alpha, which stimulate the migration of tissue dendritic cells. Thus, an immature tissue dendritic cell that binds and internalizes a gram-negative bacterium is induced to migrate to local lymphoid tissue and present bacterium-derived peptide antigens to naive T cells. Other members of the TLR family are expressed on tissue dendritic cells and are thought to be involved in detecting and signaling the presence of other classes of pathogen. Other types of receptor that can bind pathogens, such as receptors for complement or phagocytic receptors, such as the mannose receptor, are also expressed on dendritic cells and may contribute to their activation. Have a great day.